This is topic 11, structure and properties of amino acids. So this is basically our first biochemistry lecture. The topics up to this point have been in regards to chemical principles and how we apply them to biological systems. And so we'll be using those approaches as we talk about biochemistry throughout the course. So this topic will be looking at our first class of biological molecules, the amino acids, and the learning outcomes are as described. So key issues for biological macromolecules. These will always be the case regardless of the class of macromolecules that we'll be studying. First is that macromolecules usually have specific 3D shapes. These molecules can get quite large, and there are thermodynamically favorable ways and pathways in which these molecules can form stable three-dimensional structures. They occupy a given volume, and if we look at these details of these structures, they have irregular surfaces with crevices and protrusions that will allow for biomolecular recognition, interaction between two macromolecules, which is very important in signal transduction and allosteric regulation of enzymes. And we'll be talking about those in other topics. These molecules aren't a rock, they're not solid like a rock, but they do have conformational flexibility. And so when they do bind a cofactor or an allosteric effector or some smaller molecule, or even interacting with another macromolecule, there are conformational changes that occur that are a result of these non-covalent interactions. So this changes some of their properties as well, as we'll be describing in other topics. They are very dynamic. They can have certain parts of the molecule alter its shape, or it can move in a certain way and cause a active site for an enzyme, for example, to be exposed now to allow the molecule to come in. There also could be changes in temperature, which causes a conformational change, but it also could exist in an equilibrium at a given temperature, and then you actually have two conformational forms of the molecule that exist, and one may bind an allosteric effector and one may not. Then you alter the equilibrium, and then you shift it in the direction accordingly. So these are dynamic molecules. They're not static. Okay, there's not necessarily just one structure, but maybe a conformational ensemble, if you will, of different structures for a molecule. Fourth is that the function of these three-dimensional shapes of these macromolecules are important for its function, and we'll be showing many examples of that. So keep these key things in mind when we're looking at macromolecules, and we'll be talking about the details of these specific for the macromolecule that we're discussing. And it's all based on chemical principles through non-covalent associations, and sometimes it could be covalent as well. The biological macromolecule classes are shown below. There's proteins, nucleic acids, carbohydrates, and lipids. And we're going to be discussing proteins in the first part of this course, and then we'll cover nucleic acids, carbohydrates, and lipids. So the order is a little different than your textbook, but we're covering it with proteins first. And in order to understand proteins, we have to understand that they are actually polymers of amino acids. And so we need to look at the smallest structural unit first, the amino acids, and then how we connect those to make polymers, and then how these polymers fold up into three-dimensional protein structures. And so this is the approach that we'll be taking for all of these macromolecules, is we'll be looking at their smallest monomeric component, the smallest unit, and we'll be looking at the detailed chemistry of those components, and then we will work our way to more complex macromolecular structures. So the topic here we'll be looking at the amino acids. So this is a general structure of an amino acid shown right here, and notice that we have some annotations on this molecule. So we talked about amino acids before when we looked at acid-base chemistry. So it should be familiar to you that you have a carboxylate group here that has a pKa value, and an amine group that has a pKa value. And this is what we can consider acidic, and this is what we consider basic. And so under physiological pH around 7, this is how most amino acids will be looking like in terms of its overall charge as a Zwitter ion, in which there's a positive charge and a negative charge. And the R group, though, could also be charged, as we will talk about. And so therefore, its Zwitter ionic form would be at a different pH. So if we look at this molecule, the carbon that is the carboxylate carbon, the carbonyl carbon, is number 1. The carbon is attached to that is carbon number 2, or what's called the alpha carbon. So we'll often refer to it in the Greek designation. So when we say alpha carbon, we're talking about the carbon that's attached to the carboxylic acid or carboxylate group in this case, because it's deprotonated. And then what's attached to the alpha carbon is the amino group. So this is a primary amine. So this is the alpha amino group. This is a hydrogen, and you always will have that with amino acids. So this is called the alpha hydrogen because it's connected to the alpha carbon. And then you have a side chain that's attached, and it's going to be of different functional groups, and so we're just designating it as R here. There are 20 different amino acids. 19 of them have R groups that are just some kind of functional moiety that's attached, but the one amino acid that's a little different is called proline, and proline actually has a cyclic structure in which it actually has a covalent carbon-nitrogen bond to the amino group, and so that confers different properties for that amino acid, so we'll be talking about that one in detail. But they're all considered what we call the 20 amino acids. If you look at this diagram to the right, you can see it as in the ball and stick version, and the color that is shown here is usually the conventional colors for atoms, in terms of oxygens being red and nitrogens being blue. Sometimes carbons will be considered gray or black, or just sometimes even if it's a carbon hydrogen, we just consider them and color them white. It just depends if we want to denote electrostatic potential differences, where the oxygens will be and nitrogens will be one color, and conveying a more of a positive charge for the nitrogens and negative charges for the oxygens through the carboxylates, and everything else being neutral. But in this case, we have designated a alpha carbon here, designated in black, and the other carbon atoms of the amino acid is gray, and the hydrogens is white. So you'll see some variation of that as well. So just keep that in mind as you look at structures. But this is typically the convention that's used. So let's look at the more detailed structures of these amino acids. If you noticed in the previous diagram, there is stereochemistry associated with the amino acids. You see a wedge that's solid, and that wedge is sticking out of the plane of the screen, and that's going to have our side chain R. The dotted or dashed wedge is going back into the plane of the screen, and that's your hydrogen. So there is a chirality here. There is stereochemistry at this alpha carbon. The other carbon here, carbonyl carbon, That's sp2 hybridized, so it's planar. So it has a p orbital and 3 sp2 orbitals. The sp2 orbitals are participating in these sigma bonds. The p orbital is participating with a pi bond with the p orbital of this oxygen. And the lone pairs of electrons here are in sp2 orbitals. And this molecule shows also the lone pairs of electrons on the heteroatoms as well. And in terms of the plane, what's in the plane has all of these bonds here in the plane of the screen. This R group's coming out at you. This hydrogen's going back into that. So that alpha carbon is a chiral center. Now, sometimes you'll see pictures of amino acids as shown here, where the carboxylate is completely protonated and the amine is completely deprotonated. This does not exist in any solution, no matter what the pH is, and so it's better to draw these structures as they normally would exist under physiological conditions, and that's shown down here. That's the Zwitter ionic form. In this case, they're not showing the positive charge being localized to one of the oxygens, but it's showing the hybrid structure in which this negative charge is delocalized over these 3 atoms, oxygen, carbon, and oxygen. So you could draw it this way, it's fine, but you can also draw it where you have a C double bond O and then 1 sigma bond here, extra lone pair of electrons here, and O minus. That's completely fine. You can just draw one resonance structure instead of the hybrid structure, but what's shown here is the hybrid structure. So either one is fine, as long as you make sure that it has the negative charge on at pH 7. And we talked about the different properties, and we're going to look at everything in terms of acids, even though they could be considered bases. So we're going to look at everything in the protonated form. So the side chain of the carboxylate shown here, and it can lose a proton and generate the conjugate base form. And the pKa values here will vary from 2 to 3, depends on the amino acid, because this R group will have an effect on this pKa down here. So we're not going to go into the details of why that is, but it's because of those different atoms on R that will either stabilize the carboxylate more than other atoms. And so that's why those pKa values vary slightly. The amine, we can think of it as protonated as well as an acid, and it's losing a proton, but its pKa is going to be between 9 and 10. So that's why we consider that basic, because the pH has to be very high in order for this proton to be dissociated to form the conjugate base form. Notice in this case the acidic form is positive and the conjugate base form is neutral, whereas in the carboxylic acid the acidic form is neutral and the conjugate base form is negative. So you have to keep in mind that the pH can also change the effect of the charge on these species as well. So pH 7 is where we're normally going to see these, and so these are going to be the typical protonation states when we draw these structures. So now we're going to look at the amino acid stereochemistry. As mentioned previously, this alpha carbon is chiral. It has 4 substituents on it that are different, and therefore there is handedness to this molecule. R group with the solid wedge is coming out at you from the plane of the screen, and the hydrogen with the dashed wedge is going back into the screen. Everything else, all the atoms, are going to be on the plane along this sigma system. So recall what a stereoisomer is. It's a molecule with the same chemical formula, empirical formula, but different configurations, not conformations. Conformations is a rotational component where things can rotate and get in a different spatial environment. Configurations have to do with the covalent attachment of atoms, and those are fixed bonds that, unless you do a chemical reaction, will not break. So you can't change a configuration without breaking and reforming a actual chemical bond. And so these are stereoisomers. And so if we look at the amino acid example of serine, we can see that there is a mirror plane that can go between the two different forms of serine. So it's probably better to look at part B here and look at the diagram where we have the alpha carbon in the center, and we're going to have it situated in such a way that the sigma bonds on the horizontal positions are coming out of the screen. So those are solid wedges, and the vertical ones would be going back into the screen as shown here. So they rotated this one to make these two carbons be within the plane of the screen, and therefore this would just be going back. But you can imagine rotating this, because this is a tetrahedral basically, and both would be going back, both sigma bonds that are vertical back into the screen. So this would be your Fisher projection. Remember the Fisher projection from organic chemistry, and that's shown here, where you kind of draw a cross like this to simplify it. There's a chiral carbon at the center of this cross. We'll draw certain biological molecules in a similar way, especially when we go to carbohydrates. We'll have a C here, and so when you see these linear forms of sugars, you will be looking at basically a Fisher projection at each chiral center. Each carbon of that molecule will be arranged in such a way, if it's chiral, it's in a Fisher projection. So it matters what's attached to it. So if we go back to this version of the amino acid, this would be our R group here, the CH2OH that's going down. So we have our amino group, our R group, our alpha hydrogen, and our carboxylic acid group. So we can basically rearrange this by breaking this hydrogen bond, carbon hydrogen bond, and carbon nitrogen bond, switching these two substituents, and we have a new molecule. Because now the stereochemistry is different. Even though the empirical formula is the same, has the same number of atoms, they're arranged spatially different, and we broke chemical bonds and reformed them to make this happen. And so by definition, based on experimental measurements, this is the L version of the amino acid, and this is called L-serine. The ball and stick diagram is shown here. Kind of hard to see the stereochemistry there. Maybe it's better for some of you. Shown as the ball and stick diagram. And when we break these two substituents and switch them, now we have the D version of serine. L has to do with the plain polarized light that goes through a solution of a molecule such as serine, and you'll see the light will be reflected in a direction that's to the left of the detection device, and so it's called LEVO, or we have a capital, actually a small capital L to designate that, and so this would be the L version of the amino acid. And this convention is based on glyceraldehyde, which is a 3-carbon monosaccharide. You'll learn about that later, but that's where the designation came from in terms of its identifying as being an L amino acid. Okay, so that's kind of historical. So we call this the L version. So notice in the way we draw the stereochemistry in the Fisher projection is that the amino group is on the left. Okay, the carboxylic group is at 12 o'clock at the vertical position. The hydrogen is at 3 o'clock, and the R group is at 6 o'clock. So if you have this configuration here, carboxylic acid group at the top, R at the bottom, hydrogen on the right, amino on the left. So if you have amino on the left, it's L. If we switch these two, now the amino group is on the right, it's D. So that's a simple way to tell. And again, D is right because it's related to the Latin dextro. So dextro-rotary or level rotary. So it has to do with the plane polarized light measurement. So if it's the opposite mirror image, it will rotate the plane polarized light in the opposite direction. So together, these are called enantiomers because they're stereoisomers. They're mirror images of one another, so remember that term. So we got stereoisomers, and a pair of stereoisomers are enantiomers. We also have diastereomers as well, and you'll encounter that when we look at the carbohydrates. So what I want you to focus on is that L-amino acids are the ones that are prevalent in our biological systems. Now there are some Ds, and you have to have enzymes to help make those, to move these groups around differently. But the ones we have in proteins are L-amino acids. So they all follow this particular designation here, where your R group is at the bottom, the amino on the left, carboxylic acid group on the top, and the alpha hydrogen on the right. Okay. So there's some problems to work on that you have to define whether it's D or L. You have a 50-50 chance of guessing, but it's not too difficult to figure it out. As long as you have the stereochemistry in terms of what's coming out at you and what's going back, it's easy to tell if it's D or R. Now here we just shifted these two around and exchanged them into opposite positions to get the opposite stereochemistry. But if we were to switch them back, we go back this way. But you could have also switched the vertical ones around, and that would also give you a different stereochemistry. And we could switch the other two around and go back. So there's different Fisher projections that would be equivalent, depending on where these R groups and the other substituents are located within the Fisher projection. So if you work on the problem sets, you'll get a better idea of recognizing whether these are D or L. And so it just takes a little practice in doing that. But if you keep this in mind, what's L? It's always going to be this. You can always figure out if it's D or L. If you can redraw your structure to match this form. If it doesn't match this form, it's going to match the D version over here. So it's just more of keeping the carboxylic acid group on the top, R group on the bottom, and whether the amino group is on the left or the right. And that's going to tell you if it's L or D, respectively. There's also the system of looking at configuration from an absolute perspective, and that's the RS system. And that was based on prioritizing substituents. And we're not going to go into this in this course, but I just wanted you to be aware of it. Based on the different functional groups, we have a priority. And in this case, SH would be number 1, and OH would be number 2. This would get the higher priority of numbering. And you may recall when you're doing these Fisher projections in organic chemistry, you had to look at the substituents and prioritize them based on what was attached to the carbon atom. And so if they both had carbon atoms attached, you'd have to look at what's attached to the carbon atom in order to prioritize it being the highest priority or the lowest priority. Lowest priority would be 4. So that'd always be your hydrogens there. So you'd have 1, 2, 3, 4. You would have to prioritize those atoms in a Fisher projection. Then you'd have to turn number 4 away from you. Then you'd have to look at the orientation of 1, 2, and 3, whether it was in a clockwise direction, rectus, Latin for right, that would be R, or if it were left, it would be sinister, and that would be S. So I always remember sinister, like a witch is sinister, it's evil, and that's what the kind of relates to. If they were left-handed, oh, there must be a witch back in the day, and so they were sinister, and so that's how I remember S is the counterclockwise direction. But anyway, we're not going to get into that, but here's just an example to show you that if you had your L-alanine amino acid, which you have an R chain as just a methyl group, and if you look at this wedge diagram here, is it D or L? Well, it's actually L. If you were to rotate this into a Fisher projection, this would be at the 12 o'clock position, this would be at the 6 o'clock position, the hydrogen here would be on the right, and the amino group would be on the left, and so that by definition is L. If we were to prioritize these substituents, okay, the amino group would be number 1, the carboxylic acid group number 2, and the side chain would be number 3. And then the hydrogen would be number 4, and that's what you turn away from you. And then you look at the order of the substituents, and it goes from 1, 2, and 3. This is counterclockwise. This is S. And so this L amino acid, we can designate it as S in absolute configuration. This is experimental. This is absolute. These side chains usually follow the prioritization as being number 3, but sometimes that is not the case. There's a few side chains that they're prioritized as number 2, and the carboxylic acid group is number 3, and so that's going to change the priority from a counterclockwise to a clockwise position, and those are going to be R amino acids. There's only a few like that, so basically the key thing is to know is that most L's, nearly all of them, are S. But we're not going to have you do R and S. You've done that already. I just want you to be aware of that. And also, if you look closely at your amino acid structures, you may notice some of the side chains have chiral atoms in it as well. And so we're not going to go into that in terms of this course, but just be aware that there's more stereochemistry there than just in the alpha carbon of amino acids, depending on the amino acid that you're considering. So no L and D, but just be aware there's also the absolute configuration. But you will not be asked to do any absolute configuration determinations for structures. Just L or D. So the amino acid side chains, and we're going to go into the chemical details of these structures, can be prioritized based on their, or grouped I should say, based on their overall chemical properties of the side chain. And so we talked about looking at molecules and whether they're hydrophobic, have a lot of carbon-hydrogen bonds. If they're hydrophilic, have a lot of polar bonds between carbons and heteroatoms like nitrogen and oxygen. And then we also look at whether there's a charge on it based if it's an acid or a base, and the pKa of those groups and the pH of the solution. So you can have a fixed or a formal positive charge or a negative charge on the molecule. And also if the molecule is aromatic, where it has some kind of a resonance ring system as well. So we can group the amino acids in these 4 different classifications. Now these classifications aren't absolute, but these are typically the groupings that are used in biochemistry. I don't want you to memorize this. I'd rather you think about the structures of the amino acids and tell me about whether it's hydrophobic or not. And so if you think about the structure, you don't have to memorize it. You can just think about the amino acid and you'll know. So that's the best way to do it. So we'll talk about these in their structural components, whether they're hydrophobic, where they have nonpolar side chains. They're polar amino acids, that they have electronegative atoms in the side chains. They can be charged because of the fact that they have acid and acidic and basic groups on them. They can be positive or negatively charged based on the pH, or they could be aromatic ring systems. And we can classify them as aromatic, but that doesn't mean that they're necessarily hydrophobic. They could be, or they can also participate in some hydrogen bonding as well. So they do have some polar characteristics. So, but you wouldn't necessarily consider them polar but you could. There's polar aspects to it. There's also hydrophobic aspects to it. So you could stick them in hydrophobic, but they're aromatic, so they stick it here. So you can see how these classifications are just more of based on this group, but that doesn't mean it doesn't have some polar characteristics if it's aromatic. Okay, so or it's hydrophobic. It could be aromatic too. So you just have to kind of keep that. Some amino acids could be in several different categories. But if you count all these up here in their three-dimensional or a three-letter code, there's 20 of them, all right? And so how are they grouped? Based on the side chains. The chemical and physical character of each amino acid and the proteins they assemble has to do with the ability of these molecules to interact with one another based on these chemical characteristics, all right? So if I ask you, is this amino acid polar or not, I want you to know why it is. Not just say, yes, it's polar. I'll be like, okay, why is it polar? Oh, it's because it's in this list. That's not good enough. You understand the structure and why it's polar or not. That's what I want you to focus on, not memorizing lists of things. Certain things you'll have to memorize, but other things you can infer from other things that you have learned. So you don't have to memorize everything, just key things. So we'll try to emphasize that throughout this course. So here are the amino acids that you must be able to draw. So I know what you're thinking. Gee, we're going to be doing this online, take-home exams, in a sense, an open book. Gosh, I don't need to memorize this. If I have to draw a structure of these, I'll just whip this out and draw it. Yeah, you will, and it would take you a lot of time to copy it, right? But that's not going to help you when you're taking an MCAT exam, or it's not going to really help you on our exams because it's going to be timed. And if you're spending a lot of time copying out of here, that's much slower than if you draw it, okay, from scratch. You can do it much faster from memory, and you can think about the question while you're drawing it, whether if the group should be protonated or not, and how to connect these amino acids together, and how, if it's D or L. So all of the amino acids shown here are in the L configuration, in which you have your side chain as the solid wedge, and your alpha hydrogen as the dash wedged. So these groupings follow the groupings on the previous page. Nonpolar aliphatic amino acids. Here's your aromatic amino acids, your polar amino acids, and for the charged, they broke them into positively charged and then negatively charged. So everything in black here is your basic amino backbone, your carboxylic acid, your alpha hydrogen, and your amino group. Everyone has an alpha hydrogen as well, an alpha amino group, and a alpha carboxylic acid group. So the bonds in the black are the same for every amino acid, okay, except for proline here, which is connected as a cyclic side chain. So you got to keep in mind that little distinction there. You don't have 3 hydrogens on your amino, you only have 2 in this case. We're drawing these at pH 7, which these are shown. And everything else is just a side chain difference. And so we'll go through these as individual groups, and I'll highlight different chemical features that we'll see throughout the course. And we will encounter these over and over again when we're talking about proteins. But right now, the thing I want you to know is that you should be able to draw these structures on a blank piece of paper. In addition, you must know the amino acid name, the 3-letter abbreviation of the amino acid, and the single letter code, because you'll be given problems with all 3 different abbreviations in some form, and you must be able to translate that into what the amino acid should be, okay? So pretty much study this page and be able to basically reproduce it. You don't necessarily have to draw the structures with the wedges because we're going to say everything's L. So if you just put straight lines here, I'm assuming it's L. But that's what they're shown here to emphasize the stereochemistry. But unless the problem says D or L, I'm just assuming it's L. And unless the pH is different, we'll assume it's pH 7. Okay? So keep in mind when you're looking at problems, think about if it's asking for D or L and the pH, because that will affect whether these groups can be protonated or not. Okay? So this is your first biological class of molecules to really think about and study, so be able to draw these structures. It'll be very important in understanding the concepts and other topics and throughout the course. Because we'll come back to amino acids again in the last week when we look at amino acid biosynthesis and degradation. So these will come up again at the end of the course. So you want to make sure you spend time with them now and to know their structures very well. Now we're going to look at these different groupings, these classification of amino acids in a little more structural detail. So here are the aliphatic amino acids. And notice here the stereochemistry is gone. It's implied these are L amino acids. Here's your name, 3-letter code, single letter code. Notice here there's some pKa's listed. And again, this figure provides some pKa's values. Your textbook has a table of pKa values. I will provide pKa values. You can look them up on the internet, different textbooks. The numbers can vary a little bit differently. But that doesn't matter if it's 2.4 instead of 2.1 or 2.3. Just as long as you, given a pKa, you can figure out if it's protonated or not, a given pH. That's what I want you to be able to do. So this is providing that detail here, but it's just to illustrate there's pKa values associated with the amino groups and the carboxylic acid groups. But notice how some of the pKa's will change. So for example, if we look at alanine here compared to, say, isoleucine down here, you can see that the pKa of the carboxylic acid group varied a little bit. All right. Or if you look at leucine and isoleucine, you can see that the pKa of the amino group varied by 0.1. Is that experimental error, or is there some chemical reason for that based on the structure? And again, you can hypothesize it could be that based on the structural position of this methyl group here would be a difference. So there is some chemical principles underlying that distinction. But I don't want to go into those details at this point. Just know how to use these values as it relates to whether the group is charged or not under a given pH. So again, these are all drawn at pH 7. If we look at these side chains, the Greek lettering system continues. So for example, with alanine, it goes from alpha to beta to the next carbon. And then you can see for valine here, it goes from beta, the next letter is gamma, but since you have two ways to go, there's a gamma 1 and a gamma 2. Okay. If you look down at leucine, you have a gamma, but the delta you have 2, so you have a delta 1, delta 2. So sometimes you'll hear us talk about the gamma hydrogen of leucine or something like that. That's the detail that we have. We can picture it in our mind if you know the Greek lettering system of the side chains. It's not going to be an emphasis in this course, but it's just something you should be aware of in your readings and some of the topics that we may be going into. If you look at your groups here, you can just see the side chains just have more carbon-hydrogen bonds, very hydrophobic, and those are classified as being hydrophobic. They want to interact with one another, but the amino acid also has polar characteristics as well. So there's hydrophobicity, but there's also polar aspects with these molecules. Leucine and isoleucine, if you look at it, it's just a difference between a methyl group off of the gamma carbon or the methyl group off the beta carbon. So they are exactly the same. They have exactly the same mass, but the methyl group is at a different position on the side chain. All right. Also notice that this carbon at this beta position on isoleucine, a methyl group, an ethyl group, and then the rest of your amino acid and a hydrogen. It's chiral. This gamma carbon on leucine is chiral. So there is a RS designation there that we can specify. So we have two chiral centers, the alpha carbon, the gamma carbon, alpha carbon, or the beta carbon. Not going to ask you to know that, not going to ask you to draw it structurally, but if you go back in the previous table that I showed of all the amino acid structures, you can see that there's wedges, solid wedges and dash wedges there, indicating the stereochemistry. But you're not responsible to know the stereochemistry there. Just be aware the side chains do have some stereochemistry. Proline is an interesting one because it's an amino acid basically, because it's a secondary amine and it's a cyclic side chain. Notice its pK is a lot different as a secondary amine versus a primary amine. Then you don't have to memorize the value, but be able to understand what it means. And then you have phenylalanine, even though this is considered aromatic because we have basically a benzene ring attached to a beta carbon of an amino acid, but it's classified as aromatic, but it is hydrophobic. So there's that aspect of the amino acids where you could put them in either category if you wanted to, but when they consider hydrophobic in the previous grouping, it was considered aliphatic. And but in terms of its actual function in the protein, it is hydrophobic in terms of where it will interact with other hydrophobic side chains. Also notice with these amino acids, everything has a beta carbon, right? There's a beta carbon, beta carbon, beta carbon. Everything has a beta carbon, all right? So that's always going to be the case except for glycine, which just has two hydrogens on the alpha carbon. And it doesn't have a chirality because it's not a chiral center. Here's proline, a little more detailed structure, and this is important because proline, because it has this secondary amine, gives it different properties in a protein. And when we look at a protein, amino acids are connected by what's called peptide bonds, amide bonds, and we'll go into the detail of that, and we'll come back to this aspect. But this proline molecule can exist in two conformations, not configurations. Configurations is L-amino acid, but the way things can rotate around the peptide bond in a structure can be considered what we would call cis or trans. And so proline is a very unique amino acid because it's cyclic. It's a secondary amine, and that gives it unique properties in protein. So we'll talk more about proline when we get to protein structure in the other topics. The aromatic amino acids, tryptophan, tyrosine, phenylalanine. We just saw phenylalanine being very hydrophobic, but tyrosine is just the hydroxylated version of phenylalanine. So it's in the para position where the beta carbon is. So it's para-hydroxy phenylalanine, basically. Para-hydroxyl phenylalanine is tyrosine. And this amino acid has hydrophobic character, but it also has polar aspects because you have a hydroxyl group on there. It can also hydrogen bond as well. So you have a dipole moment here to make it a little more polar, but you also have hydrogen bonding capability you don't have with phenylalanine. And this one also, if you notice, has a pKa too of 10. So if the pH is above 10, that can exist in a negative charge form. Notice tryptophan is a fused aromatic ring system. It has a heteroatom as a nitrogen here. So this nitrogen is, if you extend it out, is a hydrogen bond donor. And so it can participate in hydrogen bonding. There's some polarity here. The lone pairs of electrons on this are in resonance with this ring system. So it can't act as a hydrogen bond acceptor, but it sure can act as a hydrogen bond donor. These are the things I'd like you to think about when you're looking at these structures and what you've learned from the other topics. Is this a heteroatom? Yes. Okay, there's going to be polar bonds there. Which direction will the polarity go to? Well, it's going to be pointing negatively towards the oxygen. So this will be delta plus, this will be delta minus. Does it have hydrogen bonding capabilities? Yes. Is it a donor or acceptor or both? That's what I want you to think about. All right. Notice this one, you're not going to be able to pull that proton off, so there's no pK associated with that. It's a super high value, so it's not even listed. These aromatics can absorb light, and we can take advantage of that spectroscopic characteristic and look at using this to quantify these amino acids and also proteins that have these amino acids as part of their structure. So recall the Beer-Lambert law, where absorbance is equal to the molar absorptivity times the concentration of the analyte in solution times L, which is the length of the cell. It's in your spectrophotometer, and it's usually 1 centimeter typically. And so typically the concentrations we consider molar, the cell length 1 centimeter, so your molar absorptivity would be inverse molar, inverse centimeter. So when you multiply all these out, you get an absorbance number that's an absolute number and not a value. So we can calculate the extinction or molar absorptivity constants experimentally, and we can divide them for tyrosine, tryptophan, and phenylalanine, and we can use that information to then figure out concentrations if we can measure the absorbance. And notice here in your molar absorptivity values, but for tryptophan, tyrosine, and phenylalanine, I don't want you to memorize these numbers or anything, but just understand the concept that the molar absorptivity of tryptophan is like 1,000 times more than tyrosine, which is like 1,000 times, not necessarily 1,000 times more, but quite a bit more than phenylalanine. So tryptophan is a higher molar absorptivity than tyrosine than phenylalanine. So don't worry about the numbers, but notice the fact that the more aromaticity you have, the longer the electrons can resonate over space. 2 fused rings is going to be more than 1 ring, and so its molar absorptivity is going to be higher. The difference between tyrosine and phenylalanine is this extra hydroxyl group, which can resonate with the ring, so that's a longer resonating path, and so therefore it has a higher molar absorptivity compared to phenylalanine. Tryptophan is the key one. You can have 1 tryptophan in a protein, and you can use the spectroscopic measurement to quantify it. And you would need about 10 of these tyrosines to equal 1 tryptophan in terms of its sensitivity. But together, proteins will have a multiplicity of different residues that are aromatic. It just depends on the protein. Some proteins may not even have a tryptophan in it. It just depends. But we can utilize these aromatics to quantify protein concentrations. And that's what I want you to remember is that aspect. Spectroscopic properties coming from these aromatic residues. Now let's look at the hydrophilic amino acids. We're going to look at the basic ones. Basic being defined as those that have a high pKa, which like to keep their protons. So that would be your lysine, arginine, and histidine is kind of grouped here, even though its pK is much lower than the other 2 amino acids. So it can readily give up its proton if the pH were 7, okay? The pK is 6, so that means it's going to be deprotonated at pH 7. So it's not really very basic, is it? Maybe it's more acidic. It's more acidic than these two because its pK is lower. If you compare these two, this would be the most basic, this would be second, this would be third. So that's what I want you to remember is the properties of these amino acids given these pKa's. How can you understand these functions, whether they're protonated or not under a given pH? Understanding that there's a charge associated with these when they're protonated, okay, and when they're not. So we'll see histidine a lot. Arginine is this guanidino group that's attached to the side chain, and it's resonantly stabilized, so that's why its pK is higher than the fixed charge on the amino group. Notice they're very similar structure. There's a beta, beta, beta. Like I said, pretty much the amino acids have a beta carbon except for glycine, and then you have your extension from that. So when I think of lysine, I think of it as being an epsilon amino acid. So that's how I remember to draw it. I go alpha, beta, gamma, delta, epsilon, and I stick an amino group on the end. For arginine, I think of it as a delta guanidino group, and so I go alpha, beta, gamma, delta guanidino. That's what this is. And then for histidine, it's an imidazole side chain, so I think beta imidazole. Histidine is always the tricky one. I like drawing the sigma system first, putting my nitrogens in, and then adding my double bonds in the second position, okay? So these are considered what we can call basic because they can become positively charged, even though this is kind of acidic compared to the other two. It's basic because it's positively charged when protonated. So we kind of stick it in this category. The acidic ones become negative when deprotonated. And even though you have your aspartic acid, which is just basically a beta carbon with a carboxylic acid group on it, and glutamate is just alpha, beta, gamma with a carboxylic acid group. So the difference between these two is just a methylene, a CH2. Another way to help memorize the structures. Their pKs are a little different in their side chain, but they average around 4. Okay. The difference here is because it's further away from this negative charge down here. It will have a tendency to be a little different as pKa relative to the side chain of aspartic acid. If we look at the pKa's, we can just consider them as 4 for the side chain, and their alpha amino groups, we can consider those as 2, or alpha carboxylic acid groups as 2. The side chain of cysteine is also somewhat acidic in that it will deprotonate if the pH is above 8. Its pK is around 8. So it will be S minus. It will lose the proton and become negatively charged. And so we can consider it acidic in a sense, but traditionally that's not going to be part of the acidic amino acids in that case. But it does have a pK there that would be considered acidic. Because if it's deprotonated, it's negative, like this is negative in that state. So that's why it's there in the acidic group. The unique thing about cysteines is that they can form covalent bonds between two different cysteine residues within a protein. So we'll talk more about what this stuff up here means. But right now I just want you to understand that these cysteines, and we'll get back to this a little bit later, will form these covalent, what we call disulfide bonds. So this is an oxidation event where these two negatively charged cysteines are going to lose electrons in such a way to something. Usually it's going to go to oxygen in solution, that you'll form water, and then you'll form this disulfide bond. We can also put reagents in there to facilitate this. So we can put a oxidizing agent that will take these electrons away from these two negative sulfhydryl groups and allow this disulfide bond to form. So in cells, it's mostly a reducing environment, and so it's going to favor this form, whereas on the outside of a cell it's more oxidizing, and so it's going to favor disulfide bond formations. And so we'll see that a lot in terms of proteins that reside on the outside of a cell tend to have a lot of these disulfide bonds to help make the structure rigid. Think of it as like girders in a building. It can make the building very strong and solid. These disulfide bonds do that because they're covalent bonds. They link things together. So it can make a protein very rigid and structurally sound, which is important on the outside of a cell where it's exposed to a lot of different stressors. Then the inside of the cell is protected by the lipid bilayer. So just remember that these cysteine residues can form disulfide bonds. So here's an example of how we can break those disulfide bonds with a reagent called dithiothiazole, and that's shown here. It's actually a sugar molecule. There's actually stereochemistry here, so these OHs on one side or the other are important in terms of these two being chiral carbons. And it basically has two sulfhydryl functional groups on carbon 1 and carbon 4. And basically the way it functions is that we abbreviate this as DTT, not DDT. DDT is pesticides, been very toxic and outlawed. This is DTT, and this is a reducing agent because it will help to donate electrons to break disulfide bonds. So once the disulfide bond is there, we can add this reducing agent, and it forms what's called a mixed disulfide. And eventually the DTT itself becomes oxidized DTT, oxidized dithiothiazole, and your protein cysteinyl residues become oxidized to have the free sulfhydryl. Alternatively, we could add oxidized DTT and then go back the other way. So there's ways to change the disulfide bonding of proteins, and we use this to study protein structure. So I just wanted to let you know that these disulfide bonds are important for structure, but they're also important if whether a molecule is active or not, because sometimes these disulfide bonds are important for signaling, and you may have to have a reduced disulfide for the protein to be reactive. And if it's forming a disulfide bond, it's inactive, so it's a way to control protein function. Continuing, let's look at our hydrophilic amino acids in terms of hydrogen bonding capability. So this is how we're looking at it in terms of just not being polar, but being able to hydrogen bond. Remember, we have arginine, for example, we talked about in terms of the basic group, but there's a lot of hydrogen bond donors up here with this guanidino side chain that can exist. Even though it's positively charged, it can form nice hydrogen bonds as well. So there's electrostatic interactions that can be performed with a positively charged versus, say, a negatively charged carboxylate of one of the other amino acids, but it can also participate in hydrogen bonding as well. The amino acids, asparagine and glutamine, they're basically similar to the aspartic acid and glutamic acid respectively, but instead of our carboxylic acid group, it has a carboxyamide functional group. And so this one can't lose a proton, but it can hydrogen bond. And so there's going to be hydrogen bond capabilities with these side chains. So they're polar and they're capable of hydrogen bonding. Serine has a hydroxyl group, unlike its counterpart where it has SH of cysteine. Cysteine has a pK value of 8. The serine pK, if we were to look it up, is around 18, like an alcohol. So it's not going to deprotonate under the conditions that we look at in terms of our biochemistry. around pH 7 or so. So we don't have to worry about the environment in such a way that the chemical environment is going to cause us to deprotonate. We'd have to do something extraordinary to deprotonate that. And enzyme active sites can provide environments that will facilitate that hydroxyl group to deprotonate it. But we'll talk about that mechanism, but that's an unusual case. But it's not going to happen in solution. So this will be more of a hydrogen bond donor acceptor. Here's another one. It's threonine, and so it is basically the same as serine, except the hydroxyl group is on the beta carbon, and it has a methyl group instead of a other hydrogen on the beta carbon. So this beta carbon here has two hydrogens. This has a hydrogen and a methyl group, and so often it's drawn here as this hydroxyl group, but you could draw it up at the top and just take one of these hydrogens and put a CH3 here. Notice stereochemistry associated with that beta carbon. We're not going to worry about that. So that's why you probably see it drawn a certain way to kind of emphasize the hydroxyl group and the methyl group having a chirality on this beta carbon. So it's capable of hydrogen bonding, but it's more hydrophobic than serine because it has a CH3 group instead of a other hydrogen. Tyrosine can hydrogen bond as well, and so it can be looking at being considered hydrophilic. So again, these classifications, it's aromatic. What's it over here? Well, it can hydrogen bond, and so it can interact with other polar molecules in that regard of hydrogen bonding. So I want you to keep in mind the chemical aspects of the functional groups of these enzymes. Can it hydrogen bond or not? Is it polar or not? Not too jacked up on the classifications, but based on the side chain, can you tell me what classification does this belong? Is this hydrophobic or hydrophilic? That's what I want you to be thinking about. And the problem sets kind of help you think about structures in these ways. The last slide on these classifications I wanted to kind of look at. Let's look at glycine. We didn't talk about that, but we looked at it before when we did acid-base chemistry. It's not chiral because it has two hydrogens on this carbon, so there's no wedge diagrams there. You could draw a wedge, but it's meaningless because the hydrogens are equivalent, so there's no stereochemistry there. The side chain is very small. It's just a hydrogen. It's the only one that doesn't have a beta carbon. There's a lot of what we call structural flexibility, conformational flexibility associated with this molecule in proteins, and so that's what makes it unique. It has a small side chain. There are other amino acids that we can consider amphipathic. Remember, amphipathic is a term to define whether something is two-faced. If half of it is hydrophobic, half of it's hydrophilic, for example. That's kind of what we're looking at here in terms of these molecules being part hydrophobic and hydrophilic compounds in the same molecule. So you have some hydrophobic aspect here and some hydrophilic aspect here. But you can also say the same for some other amino acids. So again, these classifications aren't absolute, but just giving you an idea of what can go on. So in this case, this sulfur is a thiol ether, basically, connected between two carbon atoms. There's two lone pairs of electrons on this sulfhydryl. It can act as a nucleophile. This can also pick up a proton in the gas phase when we do mass spectrometry analysis, for example, and cleave off there and give us a signature characteristic fragment ion that we can use to identify a methionine, for example. So there's some other unique chemistries that these amino acids have that we just really don't have time to go into. So these classifications themselves are just kind of arbitrary in a sense. They're based on some structural and chemical characteristic of the amino acid side chain, but it's not absolute because there's a lot of versatility to these biomolecules. That's what gives the versatility to how these molecules function in living systems. So it's just not unique to amino acids, but all of these biological molecules that you'll learn about. And for tryptophan, donates a proton through hydrogen bonding capabilities there. So tryptophan is abbreviated as single letter W, and so sometimes it's the first letter, methionine, met, first three letters, and then the single first letter M. Sometimes it's not, and so I think of tryptophan like this is a W here. It's like a W in there, right? So I kind of think of the side chain as a W. That's how I think of that side chain abbreviation, W, tryptophan. So to help you with the amino acids and the one-letter codes, here's something that may be useful to you. Like alanine, the first letter is A, cysteine A, and some of these have changed the names to, instead of glutamine, glutamine. So it's a Q. And then you can kind of see, here's glutamate with the E being capitalized. Again, you just have to memorize some of these. Most of it's first letter, okay? Except for phenylalanine, it's an F, not a P, because P is taken by proline, right? So we call it F for phenylalanine because of the phonetics. It sounds like an F. Lysine is a K, not an L, because L is leucine, right? And so sometimes the second letter. Or tryptophan, like I said, I like to remember that by being a much larger side chain that's aromatic and has a W kind of buried in it. So you got to know the single letter code, and this is a way that may be really helpful to you to help you memorize that. There's 26 letters in the alphabet, so 20 are represented here. One more component of amino acids is that although they have a lot of functional groups in the 20 amino acids, that's still not enough to do all of the control and regulation of protein function. So often, after a protein is made, these amino acids will get modified with a different functional group. One of the important modifications is phosphorylation. And so we'll talk about that in signal transduction because that's important for how cells communicate and cause cells to do certain gene expression and other functions within a cell for certain pathways to be activated through mediating through phosphorylation and dephosphorylation. So those that have a hydroxyl group can be phosphorylated. And so it basically now, instead of an OH group that's a neutral in a hydrogen bond donor or acceptor, now becomes a phosphorylated group. It's larger, and now it has a fixed charge on it. So that conveys a different functional aspect to the protein. Proline likes to get hydroxylated, and we'll see how that is important for some of our structural proteins. And so there's a hydroxylation specific, again at the 4 carbon, number 4, in terms of the hydroxylation reaction. And so this is going to be a stereochemical center. This is very specific, enzyme mediated. All these processes are. Acetylation of lysine residues on the amine goes from a primary amine to a secondary amine now, and it's in part of an amide functional group. That's very important when we look at epigenetics and how genes are expressed and acetylating certain lysine residues. You can also add carboxylates as well, in this case to the side chain of glutamate to become gamma carboxyglutamate. And so this is just 4 examples. You don't have to memorize these, just be aware that there are a lot of modifications that occur in these amino acid side chains that alter the structure of the amino acid, provide new chemical properties to the amino acid, which then causes the protein to have a different function. And we'll be introducing these as we go through the course when we talk about different proteins and the metabolic pathways that we'll be covering. So remember, they can be what's called post-translationally modified. After the protein has been made, enzymes will modify these residues in a controlled way to allow processes to occur within the cell when they need to occur. When you have things that are being modified and shouldn't happen, that's when we have diseases like cancer, because we can't shut signals off. So we'll be talking about examples of this throughout the other topics. But I wanted to just make the point here when we're talking about amino acids that it can even get more complicated because we can add different functional groups that changes their chemical properties and the proteins that they're attached to, their functional aspects. So to round out this topic, I'm going to talk about the acid-base properties of amino acids. And I have here that it's review because if you recall a topic on the acid and base chemistry, we talked about amino acids at that time in terms of their acidic and basic properties. So this is just more of a review to consider the fact that every amino acid has a carboxylic acid group and an amino group, and so there's pKs associated with that. We went through the different aspects of these 20 naturally occurring amino acids and had some specific pK values indicated in the structures shown in the previous slides. But on average, the pK of the carboxylic acid amino group is around 2, and the alpha amino group pK is around 9 to 10. And that the side chain could have an ionizable group that is important to consider, especially when we're looking at the functions of these amino acids. So recall the titration curve is a curve that allows us to determine pKa values, and we have equivalence of hydroxide ion on the x-axis and the pH on the y-axis, and that we start to look at how the pH changes as we have equivalence of base. So if we start a low pH and everything is protonated, and we start to titrate our carboxylic acid functional group, because that's the lowest pKa, we will have a inflection point at the half equivalence point, which will be extrapolated back to the pH y-axis value to determine the pKa. Then the pH is going to continue to rise very little until we completely titrate that proton away from that carboxylic acid functional group. And then once we completely titrate that, we're going to transition very high in pH with a little bit of base once we hit the complete equivalence point at 1, that we then begin to start to titrate the second ionizable group, which would be the amino acid group, the amino group on the alpha carbon, until we titrate that, and then we add a little more base, and then the pH shoots up. So at the halfway equivalence points, 0.5, 1.5, 2.5, whatever it is, those would be our pKa's, and that the equivalence point would be where we completely titrate that particular proton from that group. And that, recall, the isoelectric point is the pH in which the molecule is a Zwitterion, electrically neutral, equal number of positive and negative charges on it. And for amino acids just have 2 ionizable groups, the Zwitterionic form will occur between those 2 pKa's, and then the pi value is just the average of those 2 pKa values. In this case, it would be 6.1. And so this is a review of looking at that. So we can do this for every amino acid, but some amino acids have ionizable side chains, and so that will cause a difference. If you wanted to look at the species overall in terms of the pH change, we start off with the acidic form, and we titrate that away. So at the half equivalence point, we have 50% protonated, 50% deprotonated. That's the first pK value. Then we keep titrating that away till it's pretty much gone. Then we start to titrate away the other group. At the first equivalence point, we basically titrate away the acidic group, and we're just left with the basic group. And so we have this zwitterionic form that's optimal at the 1 equivalence of hydroxide ion form, and that would be, your pH would be the PI value there. Then we continue our titration, and then we start to form more of the NH2 form as we titrate that third group. So at 50% protonated, 50% deprotonated, that'd be the second pKa value. Then we can continue the titration to completely deprotonate the amino group, and so we're left with the species at the end that's completely negatively charged. So recall, low pH, overall molecular charge +1. At the first equivalence point, it's zwitterionic, a plus charge and a minus charge. And then when we completely titrate the amino group, we end up with a negatively charged amino acid. So that will be true for every amino acid except if the side chain has a group with a pKa. And here's an example of histidine. And so histidine has the side chain pKa of around 6. And so this is going to have 3 distinctive pK plateaus in the titration curve at 0.5, 1.5, and 2.5 equivalents. So where is the zwitterionic form? That's kind of what you need to be able to do. There's some problem sets that go into this. You've tried some of those already. There'll be some more when we talk about peptide and pK values there. And some more problems to do. But the idea is the same thing, is that you're looking at these transitions of different charges on the molecule based on the pH, and we're going through a complete titration curve here. The idea is, where is the PI value? That's going to be where the molecule is zwitterionic, so it's got to be at one of these equivalence points. So it's got to be at 1 or 2 or 3 for where it's zwitterionic. So you have to know what the structure is and know what the value would be if you have a completely protonated, deprotonated form, if it's zwitterionic or not. Once you find that transition equivalence point where the molecule is zwitterionic, you just average the 2 pKa values that flank that zwitterionic form. So in this case, at 2 equivalence points, it's electrically neutral, because when it starts out, it's completely protonated. It would be a +2 charge. Then we get rid of the first group, then it becomes +1 charge. Then we get rid of this group, it goes from +1 to 0, and then we start going to a negative charge. And so these are the 2 we average, because at the second equivalence point, that's where it's electrically neutral, and the PI is the average of the 2 pKa values. So shown on the left here is a table that I've posted, that you can see the carboxylic acid groups around 2, amino groups between 9 and 10. It could be a little higher depending on, like for example, proline. But notice the side chains: cysteine, tyrosine, aspartic acid, glutamic acid, lysine, arginine, and histidine. So 1, 2, 3, 4, 5, 6, 7. So 7 out of the 20, a third, have ionizable side chains that you have to worry about. So if you had to determine a PI value, this is easy. Just average these 2 because there's no side chain. But when you have 3 of them, you got to know which 2 to pick. Is it the first 2 or the second 2? It won't be the 2 on the ends. It won't be a side chain in the carboxylic acid group, but they have to be adjacent, because they have to be within an adjacent transition. So you just don't want to just pick them out of a table, but think about where the transition is. So it could be the transition where, in this case, the side chain is in the middle, but in this case, the side chain for tyrosine is higher than the amino group. But sometimes the side chain is less than the amino group. So you just can't use the values in a table, but you got to think about the structure, think about what the protonation state is at low pH, at a given pH, is it protonated or not. Those are the things I want you to be able to do. Not memorize these numbers or tables, or how to memorize how to use a table, but what do the values mean, and can you use that information to answer biological chemical problems that are provided based on amino acid chemistry, peptides and proteins, or acids and bases in general. So I want you to think about these things and to use the information, not just answer something, because I will always ask, why is that so? So you want to know why this is what it is, and understanding the chemical aspects are going to be important. So make sure you understand the acid-base chemistry here. If you're not too sure about it, because I'm not going to belabor it in the amino acid topic, but that was in the acid-base chemistry topic. So you want to go back and look at that lecture and material and those problem sets, because you'll still see this come up as we go throughout the course.